Okay. Very good afternoon, everybody. Right? Okay. So today we discuss that is the selective paper one. That is the seed technology. The final year means the sixth semester. Okay. So in this one here, the different topics also there. There is the topics here we discuss some topics are there. That is some appendages they are to discuss and harvesting and emasculation. How the rhizobium bacteria means that rhizobium bacteria are used in the nitrogen fixation. Okay, so we have to discuss one by one. Okay, see here. So what is seed? First of all, we discuss here what is seeds are there. So in this here, in any plant, you know that one, which is used for the commercial production, that can be called as a seed. So for the consumption, different purposes are there. So in the seed is nothing but the mature embryo. The seed is the mature embryo which was produced by the fertilized ovule. So in this one here, different types are there. Here, UPR seed, breeder seed, foundation seed, registered seed, certified seeds are there. Different types of the seeds are there. So based on their for the process. Another one we discussed here. So a fertilized mature ovule, which is consist embryo, stored material, and protective coats are there. That one we call as a seed coats. So this one is the very important. The seed technology means is the science deals with the method of improving physical and genetic characteristic of the seed. This one is the same seed technology nowadays. It's a very wonderful branch of the science which is used in the agriculture aspects and which is produced the different types of the crop enhancement and different varieties. You know that. So these seed technology also involves such activities as the different types of development varieties, evaluation and release the varieties. And the seed processing also, seed certification and sometimes the storage, that one is also very essential, seed storage. So now we discuss some difference between the seed and grain. So what is the seed means here? That seed is the growing plant. You know that when that natural embryo is present. And what is the grain here? We are to do the consumption. Consumption, that is the grain. The another term, another aspect is the necessary. The necessary means genetic purity and seed viability is very essential in the seed. But in the grain, there is no seed viability and genetic purity is not needed, not considered. Here in the seed, embryo is very essential. So that embryo is only to develop as the seed germinated and to develop as the seed. That is the embryo is much essential. But coming to the grain, here endosperm is essential. Why we have to consume the endosperm? Okay. So here the seed they have the certified particular company or particular variety or purity of the seed certified certification of that particular seed is essential. But the grain here no need of the certification. So the seeds also we are treated with the fungicides and pesticides for protection. But here the grain which is not treated with any fungicides and pesticides. These are some difference between the seeds and the grain also. And another term, the seed viability is also very important. So seed viability means the embryo to germinate and is affected by the number of different conditions means wholly uh, heat and pathogen different aspects also there. That's what I mentioned. And coming to the how the seed is formed. Okay. How the seed will be formed. That one means the first one is the pollination and fertilization process means the pollination means the pollen grain from one male flower to and the female stigma. The pollen grains are transferred and deposited on the stigma of the female flowers, which is germinated. And what is the seed development here? What is the seed development? Here, the process by which the seeds are formed by the end of the fertilization to produce a mature seed. That one we call as the seed development. Okay. So see here, I shown one picture. So wait some time. Few minutes wait. So we will do here. So see here. Okay. So this one is the process of fertilization. Okay. So okay, right. So this is the process of fertilization. And the structure of seed. This one is also very important. The structure of seed here. The seed of different plants may vary in many ways, but the basic structure that is the same here. What is the some parts also there? The seed coat and epicotyle and cotyledons, microphylla region. So these are the uncommon parts also there. So in the any seed which is consists these following parts are there: the testa, microphylla, and radicle. Finally, cotyledons. These are there. So what is testa? So testa means it is the outer, that is the layer for protecting of the seed. So seed coat, that one we call it seed coat, and which is the protect for the seed means embryo and other parts also. So seed coat means testa, which is the protecting part and which is outermost layer. That one we call as a seed coat also. So which is helps in the protection of embryonic plant. Coming to the micropyle. So micropylar means this one is a small opening area in this one. So in the seed, these micropylar only to pass the water for the germination time. So during the germination process, the water molecules will enter through the microphylla region. Before the seed formation, these microphylla region also enter the or allow the pollen tube. Okay. So and another one is the radicle. Radicle is the embryo root that is developed as the primary root, and the another one is the cotyledons. So cotyledons here some of the consist of high quality of starch grains also present. So this one is the food source. The cotyledons only the food source. So and the embryo will germination. The food will be supplied by these cotyledons. Okay. So here sometimes here monocotyledons also there. Dicotyledon plants also there based on their cotyledon number. So monocotyledon means non-legume plants. That is only one embryonic leaf is present. Example wheat. Cereals also there. All the cereals are coming to the monocotyledon. Wheat, bajra, and maize, paddy. These are all are the monocotyledon seeds. And dicotyledon seeds means here two embryonic leaves also there. That is the groundnut or legume plants. But all the legume plants also there. That one is the two cotyledons they have the and sometimes polycotton is also there. That is the example more than two embryonic leaf. That one we call as a polycotton. Here, example, pineapple. Pine trees means pine trees are there. So, depending on the germination here, the epigeous and hypogeous types of the cotyledons also there. That is the germination process. Yes, three types of the germination process are there: epigeal, hypogeal, and viviparous type of the germination is occurred. And the temperature required means optimum temperature here, room temperature only 25 to 30 degrees centigrade is the optimum temperature for the any seed germination. So, what is the epigeal germination here? What are the epigeal type of the germination? You can see here. The epigeal means the above of the side means the cotyledons are coming to outside of the seed, that is the epithelium. Epithelium means beans, cotton, onion. These are the cotyledons are coming out the seed and coming out of the soil. That one is the epigeal germination. Hypogeal germination means that is the below the soil means cotyledons are not appear during the germination process. They are deep in the soil and below in the soil. That is mostly monocotyledons are all the monocotyledon here hypogeal type. Okay. And another term here, viviparous germination. Seeds here thought one viviparous germination means the germination of the seed inside the fruit. That is mostly you can see in the some mangrove plants. Viviparous means the germination, seed germination inside the fruit. Example, coconut and that is mangrove plants and date palm. Tomatoes also, sometimes tomatoes also appeared inside the seeds. That is three types of the germination found there. Okay. So I show to another picture, that is the this one. So here, see here, epigeal type, hypogeal type, and viviparous type of the germination here. See here, the epigeal germination, what happened here? The cotyledons are come outside from the soil. That is the, see here. Okay, see, that is the epigea. And the hypogeal germination means that is the inside the um, below the soil. That is hypogeal. And viviparous. Tomato shows some germination. See here, these characters are it. Okay, so this one is the types of journeys. And during the germination process, here some of the external and internal factors also affect on the germination. What are the external factors? What are the internal factors? Means the factors which are influence on the germination. So external factors, water, air, temperature, light intensity. These are the external factors which are influenced. So water, this one is the very essential. And air also means oxygen for the temperature. Temperature is the light intensity. And means room temperature around 25 degrees to 30 degrees is the optimum temperature for seed germination. And another one is the internal factors. What are the internal factors? Means oxygens. So growth promoters are there. That is the seed viability and genetic material and the seed complete its resting period. These are also there some internal factors. Seed vigorous and quality. These are the very important seed viability. So how the technical yeah. Uh, how the seed that is the new technologies to improve the seeds. Here we have to discuss. So new technologies means that is the introduction of quality seeds of new varieties significantly increase yield level. This one is the very important. In India nowadays, the cultivation of high yielding varieties have helped to increase food production around 52 million tons to near 180 million tons of the 40 years, overall the 40 years. So this one is the basic tool for the secure food supplies. That is the nowadays the increase in the population. So that's why the food is very important and which is secure for the food supply. So seed improvement, that is the principal means to secure crop yield the less favorable areas of the production. Means the supply of God, that is the good quality of the seed, means good quality of the seeds which are essential for the supply. And improvement of varieties, different varieties are suitable for the areas. One of the improvement contribution to secure higher crop yields. So this one is also important. The quality of seed. This is a very essential quality of seeds. And what is that one? Yeah, and, crop improvement. These are so, and the improvement seed is the medium for rapid rehabilitation of agriculture in case of the natural disaster. So, in case of floods in the drought affected areas, the government will provide the improving seed from natural means national seed stocks, means seed banks. It is the rehabilitate agriculture product of the food grains in the country. So, what are the goals here? Also, there some goals. Some goals of the seed technology. So, here reasonable price. The seed technology to reasonable price. So, cost of the high quality seeds should be its average farmer. Means high quality of the seeds will provide the reasonable price to the farmers. It's very important. So if the normal farmer will purchase that high quality of seeds and cultivated and enhance the product. And rapid multiplication. So this is rapid multiplication through the plant breeding technology only we are involved. Rapid multiplication. And another one, timely supplies. So timely supplies means the planting schedule of farmer is not disturbed. During the favorable seasons, means the rainy season, whatever maybe winter season. Which one is the favorable for planting? During that season only, we are supply farmers. That is the one concept of this detail. And assure high quality of this. So seed technology, that is the assure the farmers to produce the high quality, high vigorous, and high improvement of seeds with produce and supplies farmers. Very essential. Means high vigorous seeds only they are made and protecting pets. It's fungal and diseases also. And another important here, the agricultural production increase. So production of agriculture means in the very short period we have to provide the different varieties we use it to agriculture. Uh, these are the some goals. Another one is some different types of the seeds also. I already discussed that one. So first one is the different types of the seeds here. Nuclear seed or breeder seed. So the first seed is also there. Only the breeder will produce breeder. The variety, which is variety, is any variety. Any variety is developed by the breeder. That can be called as a breeder seed or seed. So after breeder seed, develop the foundation seed. Okay. So after that foundation seeds means have the high quality of the seeds, bigger, then it is the all are there. Then the registers we take for the permission of the agriculture department to release the seeds to the market. And after the certification of the seeds only to give the farmers. Certified seeds that can be called as the certified seeds. And truthful. So truthful seeds also there. It's very important. So here the nuclear seeds that will be here genetic purity here 100% of purity is present. That one only produces breeder that one that's why we call it as a breeder seed. Okay. And another one is the foundation seed. So which is the color the tag of color here white in color. Foundation seed here purity of the seed here 99.5 percentage. That is the purity of seeds. And registered seeds also there. And truthful seeds. Here, truthful seeds that can be called the foundation seeds only. Truthful seeds and certified seeds. These are coming from the foundation seeds. The registered seeds that have color here purple, certified seeds blue color, and truthful seeds green in color. The tag here green color is there. And truthful seeds, the purity, genetic purity here 99% of the purity is there. That one is so see here, that is the tags I will show to. So see here, that is the tag of the different types of the varieties means register means white in color and blue color, yellow color. Hmm? and truthful seeds here, green in color, different colors. Okay, these are the different types of seeds. So generation systems of seed multiplication here, that is the particular class of seed from the specific class of seed up to certified seed storage. That is the seed stage. Basically, it depends upon the demands here. Based on the factors, the different seed multiplication models may be derived from each crop. So first of all, the seed multiplication agency should be decide how quickly the farmers can be supplied with the seed of new varieties which is released after the nuclear seed stock and handed over to the concerned agency. If it may be replaced with the old varieties also. The given basic factors, the chain of seed multiplication model to be in India third generation nowadays, in the third seed production is there. That is the this is the brief introduction of the seed. Uh,